Hello everyone. Here I am with part 5 of animal cell culture and applications. Having done part 1 to part 4, whatever topics have not been covered so far have been covered in part 5 of animal cell culture and applications. Key areas in this video lesson: stem cells, types of stem cells, applications of stem cells, creation of chimera mice, and tissue engineering. Stem cells, a cell that has the ability to continuously divide and differentiate into various other kinds of cells or tissues. It can change to one or several different cell types under proper conditions. It can undergo unlimited cell division. Stem cell research established by Ernest McCullough and James Still. Types of stem cells. There are two types of stem cells: embryonic stem cells and adult stem cells. Embryonic stem cells, also called pluripotent stem cells, are capable of developing into all the cell types of the body. James Thomson developed technique to isolate and grow human embryonic stem cells in culture. Human embryonic stem cells are derived from the inner cell mass of blastocyst, grown with the help of specific growth factors. Stem cells can self-renew without differentiation, maintain stable euplite karyotype, could be used to create chimera, and reintegrate into embryogenesis if transferred, and it retains the characteristics of founder cells even after prolonged culture. Adult stem cells. It is found in adult tissues. They are less versatile and more difficult to identify, isolate, and purify. They are multipotent, which means lineage restricted, and it acts as repair system for the body. Hematopoiesis occurs in spleen and bone marrow. Hematopoiesis is formation of blood cells, and 100,000 stem cells can produce 1 billion platelet cells, 1 billion B cells, etc. per kilogram body weight per day. Applications. It is useful in medical conditions where cells are dead or injured, like leukemia, heart disease, paralysis, burns, etc. Overview of early development. The fertilized eggs start dividing. It becomes 1 becomes 2, 2 becomes 4, 4 becomes 8, 8 becomes 16. All these stages are totipotent stem cells, which means it can become any cell in body or placenta. Then it becomes 16 cell stage, which is the blastula. In blastula, the cells are pluripotent stem cells, which means they become 3 to 5 days old. And inner cell mass of blastula contribute for the formation of the embryonic stem cells. And they are pluripotent. It can also become any cell in the body. Later, when it gets implanted in the uterus, it becomes multipotent because gastrulation begins there in day 14, and it leads to the formation of the primary germ cells. So you have formation of 3 layers: outer layer, which is called ectoderm, middle layer, which we call as mesoderm, and inner layer, which we call as endoderm. Multipotent cells can become any cell within a specific germ layer or cell lineage. So totipotent stem cells becomes pluripotent embryonal stem cells, and that becomes adult multipotent stem cells. And here pluripotent stem cells can become either somatic stem cell or germ stem cell. Germ stem cell gives rise to gametes, whereas somatic stem cell gives rise to the somatic cells. Here again, it's another picture where the in vivo fertilized eggs, which is totipotent in the 8-cell embryo stage, it becomes 16-cell blastula, and from the blastula, the blastocyst is shown here. The inner cell mass is taken out. They are the embryonal stem cells, and it is pluripotent, and it can also become any type of cell. It can become blood cell, it can become cardiac muscle, it can become neural cell. Possible uses of stem cell technology. It can replace tissues or organs. Cell replacement therapies. Cells could be stimulated to develop into specialized cells that represent renewable sources of cells and tissues for transplantation. It can also treat injuries and various genetic and degenerative conditions, including muscular dystrophies, retinal degeneration, Alzheimer's disease, etc. It can be used in the delivery of genetic therapies and chemotherapeutic agents. Next, let's look at the gene knockout. Precise modifications in the mouse embryonic stem cells and creating mouse models of human diseases. And it's extremely useful in understanding genetic basis of disease in new diagnostics and therapeutics. So here you can see the blastocyst, and the inner cell mass of the cells is taken out. So here you have the embryonic stem cells. Using electroporation, the new genes have been introduced into these embryonic stem cells. So here we select for those cells which have got the integrated new gene of our interest. That will be selection with resistance marker, and then those cells will be reinserted, those embryonic cells will be reinserted into the blastocyst. It will be mixed up with the existing embryonic stem cells. Creation of transgenic and chimeric mice. So here there are three scenarios. In A, you have the donor female mice. From there, if you take the mouse embryo at 8-cell stage, which is totipotent, then we are inserting a new transgene through retrovirus, and that will be inserted into the body of a female mice, and the offspring are screened for the presence of the transgene. In the case B, you have again donor female mice. Here, the DNA is injected into the male pronucleus of the fertilized oocyte. So when the fertilization happens, then the fertilized oocyte, injected oocytes, or in vitro cultured early mouse embryos. When it divides, it becomes the blastocyst. After it becomes the 16-cell stage, then that embryonic stem cells is implanted into the pseudo-pregnant mice, and offsprings are screened for the presence of the transgene. In the third case, embryonic stem cells, DNA with targeted transgene is introduced into the embryonic stem cells, and they are selected and expanded. Targeted embryonic stem cells are injected into early mouse embryo. An embryo is implanted into pseudo-pregnant mice, and chimeric offspring are mated with the wild-type mouse, and breeding and selection to the homozygosity of the mutation. Homozygous are where you have the same alleles. So here we have three different ways. In one we have used retrovirus, in another we have used microinjection, in another we have used recombinant DNA technology using a plasmid and a targeted transgene and introduced into the embryonic stem cells to create chimeras. Tissue engineering. Supplying body parts for repair of damaged tissue or organs without causing an immune response or infection or mutilation is called tissue engineering. Naturally derived or synthetic materials, it may be engineered into scaffolds. When implanted into the body, could act as a template. And that allows the body cells to grow and form new tissues. And they function as new organs in patients without triggering the immune response. Advantages of tissue engineering are dramatic improvements in low-cost medical care. Large-scale culturing of human or animal cells may provide substitutes to replace damaged components in humans. So here in the picture, you can see the basic principles of tissue engineering. So from the humans, we take the cells from a biopsy. You culture it. You have the monolayer animal cell culture. You expand the cells, and these cells are cultured on a 3D polymeric scaffold which act as a support. And then these cells will start growing on it and forms the tissues, and we have the generation of a graft, and that will be inserted back into the human. Thank you so much for watching this video. If you like this video, do click the like button and subscribe to my channel if you have not, for many upcoming biotechnology related videos. Thank you so much once again.